Hey guys, so this is our last video for chapter 2. It's on staining our specimens. So the first thing we mentioned before when we had Cindy and her wound on her knee is that they had taken the sample and they just did a straight up wet mount. So a wet mount was where I was telling you about. You got your glass slide, you take your sample, you smear it on there, you add some water to it, and then you pop on a cover slip, and that's your wet mount. Um, some of us have done this in previous biology classes where we took the Alodia leaf and you ripped off the leaf and you put it on your slide, you put on some water, cover slipped it, and you stick it underneath the microscope. And then I think we were asked to check out the cells, count how many cells there are in the plant cell, look at the chloroplast and all that stuff. But you put your sample on a slide, water, cover slip, it's a wet mount. Some things we all need to fix and preserve it. So you need to undergo fixation. So some things, if we're talking about tissues, if we want to see multiple cells, we need to see a tissue, we want to preserve that tissue, we're going to put it in some sort of fixing agent, like formaldehyde or something like that, to preserve the tissues, to stop the breakdown of it and keep it as is. So we could undergo this with entire tissues, excuse me, or if we've got a culture of little teeny tiny guys, individuals, bacteria growing in a culture, and we want to look at the bacterial growth, then we're going to take that sample on your from your collection, from your culture, and you're going to scoop it up, and you got your glass slide, and you got your loop. I'm pretending this is my loop that I have sterilized, right? I heat sterilized it in the Baxinator, pulled it out, let it cool, and then I have a sample in a petri dish of something that I've cultured. So I'm going to scoop up that sample and put my petri dish away, cover it up, make sure nothing else gets in it. My sample's on my loop. And then I have my glass slide, and I'm going to take that and I'm going to put it on here. So I got this fresh sample of culture of whatever this mysterious stuff is, and I'm going to put it on my slide, and then I'm going to burn all the stuff off my loop, and I'm going to put my loop away. And now this glass slide has a sample on it, but I want to heat fix it. So you're going to take that glass slide and you're going to put it in front of your Baxinator like so, and wave it in front of there, and it will dry your sample and heat fix it, stick it to the glass, and then you can stain it. So there are different stains. There are simple stains, like if you have done this cheek cell swab where I was telling you about, you go and you take the toothpick and you kind of swab some cells off of there, and then you put it on your thing. You need to stain it so you can have some contrast, so you could see these individual cells right here with the BIOL fat nucleus. And we would use a simple stain like methylene blue, something like that, to accent your cells. But everything is all one single dye, one color when you're doing this. Or instead of a simple stain, you can do a differential stain. And differential stains are going to use more than one color, more than one staining, and that's going to help you distinguish between two different types of organisms, because a simple stain wouldn't be able to differentiate between. If you use two different colored ones, it's a differential staining technique, and you'll be able to tell the difference between different things if you do that. On the stains, on those dyes, they could be called a basic dye or an acid dye. Basic dye, the chromophore that has the coloring agent, the coloring pigment. These dyes are salts. And so if the chromophore is going to be the positive part of the ion, then it's called a basic dye. If the coloring ion, the chromophore, is the negative part, the anion, then it's going to be called an acid dye. When we stain things, we can stain the object itself, the organism itself, just like we were looking at these guys with the cheek cells, and we stained it, and the blue is absorbed in the cells itself. That would be a positive stain. Here's another example of a positive stain where the cells, the bacteria, absorb the stain, and then what's in the field around it is bright field, and it's clear, and that doesn't have a stain. Or you do another type, and it is negative stain, and that's where the organism itself isn't absorbing the stain, but the background, your field, is absorbing the stain. So everything's dark. It's like bright field and dark field. Your field is dark on a negative stain and your organism is bright. And then positive is just the opposite. Your organism is dark on a brighter field background. So, excuse me, simple, one stain, just one color, everything is one color. Differentials are where you use two different types to differentiate between one thing or another thing. One type of differentiating stain, again, is gram staining. You will need to know gram staining. We do it in face-to-face lab. We do it in Labster. So you will need to know the steps. So let's go through the steps for the gram stain. So your very first thing you're going to do is use your primary stain, and that's going to be crystal violet. And it is a beautiful violet purple. And you are going to, after you've heat fixed your sample, right, you're going to put your crystal violet on there. And you're going to leave it, and you leave it for 1 minute, and that crystal violet is going to absorb into the cell walls. Now remember, we talked about bacteria have cell walls that have peptidoglycan in it, and peptidoglycan may be very thick or very thin. If it is a gram-positive bacteria, it has lots of peptidoglycan, and it will absorb that crystal violet into the cell walls. What makes it stick and stay in there is going to be, okay, so a minute has passed, we're pretending a minute has passed, and then we're going to rinse off our crystal violet. We're going to add in iodine, and iodine is going to be what fixes that stuff in. So it's a mordant, it's going to make that dye less soluble, and makes it stick to the cell walls, and that's going to sit for a minute. And then we're going to rinse it real fast with some water. Then we are going to use alcohol. The alcohol is your decolorizer. So with this is like your most important step in your gram stain, is the decolorizer. You don't want to over decolorize your organism, because otherwise that purple, if it's exposed to too much alcohol, too much ETOH, that alcohol will decolorize the purple, and it will make the purple fade, and your purple starts to look like it's pink, and that's not what you want. So you only want to decolorize for a very short time. So you will have your slide, and you've got your dye trays underneath to catch all the drippings and all of that stuff. So you have your slide, and you've put on your crystal violet, you've put on your mordant of iodine to lock in the crystal violet into the bacterial cell walls, and then you're going to drip very carefully your decolorizing alcohol, and you will watch it go down the slide. Here is your slide, and here is all of your organisms up here, stuck heat fixed to your slide, and you have stained it with purple. And so as you are dripping drops of your alcohol onto your slide, it's going to accumulate down here, and then it's going to drip off. So as these drops start coming off, they're going to be purple at first, but then as you continue to drip down, they're going to go clear. As soon as you see those drops going clear, then you're going to stop the alcohol. You stop that, and it's only, again, going to take just a couple seconds for this to happen. So you stop decolorizing right away, and then you want to rinse it with water, just like we've done in between all of these steps, and rinse that off, and that's going to stop the decoloration from happening. Because if you keep that alcohol on there, again, the longer the alcohol is on there, the longer it's going to fade your crystal violet, and then it doesn't look violet anymore. It looks reddish pink, and you don't want that. So we have done crystal violet, step number 1. We did iodine, that's our mordant that locks in the crystal violet. Then we decolorized, step number 3. That decolorizer is going to pull out the purple stain if there's not a whole lot of peptidoglycan. And what it replaces it with is going to be our very last step, and that is going to be the pink colored dye, safranin. So this is called a counter stain. Our crystal violet was our primary stain. Safranin is going to be our secondary counter stain. And so it's going to, again, it's going to be a pink color. We're going to leave that on for 2 minutes. And then after 2 minutes, we're going to rinse that with our water. And then we get to throw it under the microscope and look and see what we have. If it is gram positive, it will look purple. If it is gram negative, it's going to be pink because we washed off the purple when we decolorized because there's very little peptidoglycan to hold that crystal violet, and when it gets decolorized, it goes away, it gets washed away, and then we replace it with the pink safranin. If we have gram positives, that pink safranin's not going to stick because we already put the crystal violet, and that's going to override the other stuff. So gram positives, purple. Gram negatives, pink. Know your steps to gram staining. They're in the textbook, they're in the outline. You will do them in Labster, and you will have a few questions on a test on gram staining. So let me know if you have questions about gram staining. Here is a really great picture of a gram stain. You can see it is a differential stain, right? You have mixed organisms in this slide. You have two different kinds, at least in here, and you can see some are pink and some are purple. So again, the pink stuff, gram negative. The gram positive stuff is purple. It is positively purple with peptidoglycan. All the P's are in there for gram positive purple peptidoglycan from the crystal violet sticking into those cell walls. So this is a differential type of stain. You will need to know the steps of gram staining. Okay, so let's update. We said Cindy had the scrape. They sent it off. They did, what did they do? They did a light microscope, and they did it on dark field last time, and they discovered that there are cells there and that they were coccyx shaped and that they were in clusters. So we said that that tells us stuff. What's the shape of the bacteria and how does it grow? How is its arrangement? Is it in clusters? Is it in chains? Is it in individuals, or is it in a group? How does it grow? So we have a little bit of knowledge. And so now that we know that there's something there and it's got a shape, let's take a little bit of a better look at it. So after we did the dark field and we figured out some clues about it, now we're going to come back and we're going to gram stain it. And that gram stain, again, is going to differentiate. Is it gram positive or gram negative? Does it have peptidoglycan or does it have very little peptidoglycan? Gram positive and gram negatives are differentiators in the microbiology field. So they did a gram stain, and here is the picture of it, and they are purple. So purple is what? Gram positive or gram negative? It is positive, positively purple, all the P's. So this one is gram positive, and remember, it's positive because it has lots of peptidoglycan in the cell walls. Moving on. Another type of stain is acid-fast stain. If we were face-to-face, you'd be able to play around with acid-fast stain. It's another type of differentiator. The two dyes, instead of crystal violet and safranin, these are going to use methylene blue and carbofusion. Carbofusion is going to be the stuff that sticks. If the cell is going to have mycolic acid in it, it's kind of waxy stuff in its walls, then it will absorb the carbofusion fuchsia colored stain into the walls, and it will keep that. If it doesn't have the mycolic acid, it won't absorb the carbofusion, and then your secondary stain, your methylene blue, everything else will be blue. It'll be blue. It only keeps the carbofusion fuchsia color if it's got acid, the mycolic acid in its cell walls. So this is an acid-fast stain, and if we were looking for something like TB, it has mycolic acid in its cell walls. So this is a way that we can differentiate and see, do we have TB or not? So this is acid-fast stain. I'm probably not going to ask you steps about acid-fast, but I do want you to know about it and what specifically it is looking for in the cell walls, and that is mycolic acid. And it's going to use carbofusion, this beautiful fuchsia color contrasted against methylene blue background for the stuff that don't have the mycolic acid. Another thing that we might want to look at is, does it have a capsule? If it has a capsule, it's more virulent. So if you are a bacteria and you are pathogenic and you've got a capsule, that allows you to stick on and form a biofilm, stick on to I always go to plaque. Plaque is a biofilm. I just actually had my teeth cleaned today. So I went to the dentist today and had my teeth cleaned every 6 months. And so she scraped away all the biofilm and cleaned it all away and got it all nice and healthy teeth again. So we got rid of that biofilm. But a way for the bacteria to reattach themselves and stick onto things and onto surfaces in your body is if it has a capsule. If it's got a capsule, it's more pathogenic. It helps it be more of a bad guy than one that didn't have a capsule. So we can look and see, does it have a capsule or not? Is this one of the bad ones? Or, maybe it's an E. coli that doesn't have a capsule on it. Whatever it is, we can look and see. So we'll do a negative staining technique here. On this one, they're using India ink to darken the background, and then the organism doesn't have a halo. If it has a halo, it's got a capsule. If it doesn't, then it'll just go straight to that organism, and you won't see this glowing halo around the organism. So we have halos on both of these, this dark India ink, and then we got halos here, and then we have, over here, we had crystal violet, and see these clear, see how it kind of looks like it glows right here in this area? These have capsules. So which organism is this one? This is Bacillus. It's one that we use in our face-to-face lab. We use B sub, and so maybe we'd be able to look and see if our B sub has a capsule or not by using this staining technique. Oh, excuse me, sorry. What if it uses endospores? What if it's reproducing and it's going to make endospores? So it is going to be able to put that endospore out in its environment, and when things get better, that spore is going to grow and turn into a vegetative state and new bacteria come alive. So is it producing endospores? B subs make endospores. So we may want to check that out. One of the things that you do for endospore staining is to use a different color dye, and this one is green dye, and it is from malachite green, and you do that one first. So you're going to do malachite green, and if there's an endospore, the endospore absorbs the green. If it doesn't have endospore, it won't absorb the green, and when you wash it away, it's going to wash away. Then your secondary stain after that is going to be safranin, which we know is pink. So if we have both pink and green, then we know that these guys have endospores in their bacteria cells. So that helps you know that, you're looking at this bacteria versus a different bacteria that maybe doesn't produce endospores. What if we wanted to know about if this one has flagella or not, and how many flagella? Is it just on one end? Is it covered? Is it on both ends? That's a differentiator among different types of bacteria. So if we wanted to, we could stain the flagella with a flagella stain. It's going to have a mordant as well, similar to the gram stains that help cement in that pigment into the flagella. So simple stains, just, like the one simple thing, not differentiating, just one thing. So we've got basic ones, acid stains, and negative stains where the background is dark and the organism is positive. Differentiating when you have two different colors, pink or purple, blue or fuchsia, on our differentiating, green or safranin. Oh, I have lost my Bluetooth. Let me go back. Okay. So here is our simple stain table that I was just telling you about. The simple stains, basic, acid, negative stain, and differentiating stains where there are two different colors, pink and purple for our grams, fuchsia and blue for our acid fasts, endospores where green and safranin for these guys. It could be flagellar stains, it could be capsule stains. These are differentials. The last bit of stuff I think we have on here is the electron microscopy. I'm not going to ask you how they prepare that. It's a little more complicated, but it's going to take your tissue and embed it in some plastic resin, dehydrate it, use some alcohol, and then slice it really, really thin and then put it in the machine with different types of substances and fire off the electrons on this thing to see what it's going to look like. So this is a picture of how they prepare the specimens for electron microscopy. And then we already talked about to stain them or not to stain them. Maybe you just do dark field microscopy so you can look at it under, you can look at it while it's still alive, because some of these guys don't stain well. Some of them don't take stain. Some of them, just the process of heat fixing them and staining them just is not what you want for your organism. So sometimes it's best to keep it alive and look at it in its natural state. And so we need to do some of those different techniques that we talked about on all of those different microscopes are able to do. Sometimes we might do dark field, sometimes we might do bright field. It just depends on what you've got. Sometimes we might do fluorescence if we were looking for, here's our fluorochrome. If we were looking for, this is an immunofluorescent assay, is this the proper organism? And we're going to coat it with antibodies. And if it is the bright organism, those antibodies are going to stick to it. And if we've got a fluorochrome attached to those antibodies, then it's going to glow, and we're going to see that organism covered in the antibodies. All right, so what happens with Cindy? We're at the end of our chapter, and we need to know what's going on with Cindy. So based on what we know so far, Cindy had an infection. It was some spherical cocci bacteria. They were gram-positive. They were growing in grape-like clusters. And so we know that that's typical of staph. So they're going to run some tests and see if it is drug resistant. If it is MRSA, is it Staph aureus that is drug resistant? So they're going to run some tests for different types of antibiotics, see how it does with the specific ones coming from Cindy. And they tested lots of different ones, and they found the particular strain of Staph aureus that Cindy's got. They're going to give her the proper medication and make sure that she takes all of her antibiotics, because guys, drug resistance is real. You got to take all your antibiotics, and it clears up. So she is good to go, and problem was solved. So Cindy had Staph aureus, and it was out of control. They got her the proper antibiotics, and now she is all better. So we finally had a good resolution for chapter 2. Yay! If you have questions about chapter 2 content, please shoot me an email, or we could do a Zoom meeting, whatever you want to do. Just let me know. Thanks.